Good morning, everyone, and welcome to this MDUK seminar on FSHD. We've just gone live and I can see the participant numbers still rising, so we'll be starting in a couple of seconds once I see that number tail off. I'll just give people a couple more seconds to join. Fantastic. So welcome to this morning's Muscles Matter 2021 online seminar on FSHD, which we're hosting at MDUK. I'm Rob Burley. I'm MDUK's Director of Campaigns, Care and Support, and it's a real pleasure to be joining you for the latest seminar in this series. We've got a really packed agenda this morning with a fantastic array of speakers, and we're going to split the session into two parts. So we'll begin with looking at the latest developments around research into FSHD, and there'll be a chance to ask any research specific questions at that point as well. We'll take a 5-minute comfort break at around 11:50 because we have got quite an intense agenda, at which point we'll move on after that break to discuss living with FSHD, where we'll focus primarily on answering questions that you've been submitting in advance or that you can submit throughout the course of the session. You should be able to see the Q&A function at the bottom of your screen, which we're using for questions and comments. Please type your question or comment and we'll feed them into the discussion to the panelists. To help us manage the technical side of the event, we won't be calling on questioners to ask their questions directly. And any questions we don't manage to answer or don't know the answers to, we'll seek to answer through our website afterwards. We're going to end the session today with some information about an initiative called UK FSHD. So before we begin, a small bit of housekeeping, and note that our helpline is available to anyone affected by muscle wasting conditions, and do please contact us if you have any questions or are in need of support. We can help you with information and can either support you directly or point you to where to get the support you need. The number and email address should be on the screen now. You can call us on 0800 652 6352 or email info@musculardystrophyuk.org. And a final reminder that we're recording this session and it will be made available over the next few days. So that's enough from me for now. I'm going to start us off by introducing my colleague, Dr. Kate Adcock, our Director of Research and Innovation, who will introduce our research speakers and then chair the first part of this morning's session. Kate, over to you.
Thank you. Thanks very much, Rob. And good morning, everyone. I'm really delighted to introduce a truly fantastic panel of speakers who've joined us this morning. We'll be hearing from so many fantastic people, starting with Professor Peter Zammit from the King's College London. He's a Professor of Cell Biology there. He'll be followed by Dr. Linda Popplewell, who's a reader in molecular therapies at the Royal Holloway, University of London. She'll be followed by Dr. James Lilliker, who's from the University of Manchester, where he's a senior clinical lecturer, and he's also a consultant neurologist at Salford Royal NHS Foundation Trust. He'll be followed by Dr. Enrico Bugiardini, who is a clinical research associate at the Department of Neuromuscular Diseases at University College London and consultant neurologist at the National Hospital for Neurology and Neurosurgery in London. And then finally, we will have a video from Ben Porter, who is the curator of the UK Myotonic Dystrophy and FSHD registries, who's based at Newcastle University. Ben can't be with us today, but we are joined by Lindsay Murphy, who will be able to answer any of your questions that you might have regarding the registry. So without further ado, because I know we've got a lot to get through this morning, I'm going to hand over to Professor Zammit, who's going to tell us about DUX4 and other factors that contribute to muscle wasting and muscle weakness in FSHD. So Peter, over to you, and we'll have the slides driven by Kat.
Okay, thank you. First slide, please. Do my Chris Whitty. So basically, this talk is going to start with very basics of cell biology and DNA, and then just build up to what causes FSHD before we get on to a bit of the research. So if you see on the next slide, obviously the human body is made up of cells. You're made up of 37 times lots of zeros cells, and each cell essentially is a little factory. It's got a nucleus that is the office that controls it. It has the cytoplasm where things occur. Next slide, please. And so how you get different tissues are collections of different cells that work together. So obviously muscle cells are there to make you move, make muscle contract, but obviously lots of different specialized cells. And what makes one cell different from another cell is the proteins they make. So muscle cells make muscle proteins, skin cells make skin proteins. Some proteins are in common, but there's these specific proteins that make a particular cell different from another cell and gives it the different function, a hair cell versus a muscle cell. Next slide, please. So if we think of that analogy where we have the office and the factory, so in the office, we have the instruction manuals. These are called chromosomes. So these tell the cell all the potential proteins it can make. Okay, next slide, please. And this is what you see when you kind of see these genetic descriptors. You have all the pairs of the chromosomes. So there's 46 chromosomes in a person, and they are important for containing all the genetic information you need to make different proteins. Next slide, please. So therefore, we have all these chromosomes. So every single cell has the instruction to make every single protein. And these chromosomes are made up of genes. And genes are individual descriptors to make a particular protein. So if you want to make protein X, there is a gene for protein X that is on a chromosome somewhere. Okay, next slide, please. And so we have the cell. In the nucleus are the chromosomes, and the chromosomes are made up of DNA. So lots of DNA, it's a linear strand of DNA that is millions, billions of units long. So this is what makes chromosomes. And this DNA is divided up into genes. So you take this big linear sequence, and then it's just chopped up into sections, and the first section makes protein X, the next one makes protein Y, the next one makes protein Z, and each gene encodes for a protein. Next slide, please. And so the cell photocopies the genes it wants. So it doesn't want to make all of them. It only wants to make the ones that it needs. The muscle cell just wants to make the muscle proteins. Okay, so it copies certain genes. And these genes are copied from the DNA to RNA. Next slide, please. So we have our DNA. You copy the DNA to make RNA. So you're copying the sequence of the bases in the DNA, and you use that RNA as a code to make proteins. And the order that the bases are in tells the cell which order to put in the amino acids that together make a protein. Next slide, please. So we have our chromosomes full of DNA. The DNA is divided up into genes. The cell chooses which genes to copy. It makes a copy of the DNA into RNA, and then the RNA goes into the cytoplasm, and in the cytoplasm, it encodes for a protein. Next slide, please. As I commented, the DNA is in a certain order. It's made up of 4 separate bases. And so when it's copied, the RNA has 4 separate bases. And the order of those 4 bases tells the cell which amino acid to put in which place. So therefore, you can get different proteins because you have lots of different amino acids. So the order that you put your amino acids in dictates what the protein's function will be. And because you've got 20 plus amino acids, there's lots of variation. So as you read the RNA, you see that if you have a GCU in that order, then you will put an alanine, an amino acid called an alanine in. If it's then followed by an ACG, as this example, you put a threonine in, and so on and so on. So you can then make a protein with lots of different amino acids that gives it different functions. Next slide, please. And so this is exactly what's happening here. The cytoplasm proteins in the cytoplasm are reading this code, and by the order that the code is in, it tells the cell which order to put the amino acids in. You join the amino acids into a long string, and all those amino acids together make a protein. And that protein is going to have different functions depending on which order you put the amino acids in, which is dependent on which order the DNA is in. So the DNA dictates what amino acids go in which order, which dictates what the protein's functions are, and therefore the protein can have different functions such as in muscle, in hair, skin, etc. Next slide, please. So this is fine. The only thing we need to remember is that a hair cell doesn't want to make muscle genes. A muscle cell doesn't want to make hair genes. And so you have to turn certain genes off. So a cell is only going to make the genes it wants. It's not going to make every single gene, even though it's got the information in the chromosome. It has all the information to make every single protein in the body. Every gene is there, but it doesn't want to make all of them. It only wants to make some of them. So you turn off the genes you don't want. And you do this by various ways. But for this example, methylation. You just do this process called methylation. You methylate the gene and then the gene's turned off. So the cell doesn't make it. It just makes the ones it needs. Next slide, please. So in FSHD, next slide, please. We have, at the end of chromosome 4, so as it's commented, you have all these 23 chromosomes. On one of them, chromosome 4, at the very end of chromosome 4, there are lots of these little units that are repeated. They're stretches of DNA about 3,500 bases long that are just repeated, one after the other. So lots of these, they're called D4Z4 units. They're just lots of them stuck on in series at the end of chromosome 4. And because the body recognizes that they are repeated sections of DNA, it knows to turn them off. So it methylates them so that you cannot make proteins from that region of DNA. It's locked by being in a series of repeats. Next slide, please. But what happens in FSHD, for some reason, is that you have fewer of these D4Z4 units. So in the normal, healthy, controlled population, then you have 10 to 100, 150 of these repeats. In FSHD, you have less than 10. And when you have less than 10, the body doesn't methylate, doesn't turn off that region anymore because it's too few. It only recognizes lots and lots and lots of repeats. When it sees lots of repeats, it turns it off. When there's only less than 10, it doesn't recognize it as a region that should be turned off. So it's left on, and therefore it's not repressed. Next slide, please. And because of that, the cell, the muscle cell, recognizes it as something that it should be making. So it makes the protein from those D4Z4 units. But the problem is the protein it makes is called DUX4, and that is a protein that shouldn't be in muscle. So suddenly you have a protein in muscle that shouldn't be there. That's not good. But the other problem is that DUX4 is also a transcription factor, so it controls other genes. So you're creating a protein that shouldn't be in the muscle cell that then turns on other genes that shouldn't be in the muscle cell. So you have this cascade of problems by having this gene, this protein present. Next slide, please. And if you look at some cells from FSHD patients, you can occasionally see DUX4 present. It's quite hard to detect, but you can see it in certain situations. So you have the presence of a protein that shouldn't be there. And that protein is able to control other genes that shouldn't be there. So it amplifies the effects by controlling other genes. And together, these upset the carefully coordinated gene expression in a muscle cell by making proteins that shouldn't be there expressed, which then upsets the muscle's function, the cell's function. Next slide, please. Next slide, please. So on the left is a piece of muscle from an unaffected individual, and you can see that it has muscle fibers. These are muscle fibers. They're coming out of the screen towards you. You're looking end on, a bit like a length of hose pipe. The muscle fiber is a long cylindrical cell, so it looks a bit like a piece of hose pipe, and you're looking end on to down the hose pipe. And that's what you see. So you've got regular sized muscle fibers, and you've got nuclei on the edge of those muscle fibers. Whereas if you look at a piece of muscle from an FSHD patient, you can see that you have certain changes. The muscle fibers tend to be different in size. You have some inflammation going on, some inflammatory cells invading the muscle, and you have some muscles that are degenerating. So these are all signs of what is called dystrophic muscle, FSHD type muscle. You have scar tissue, lots of fibroblasts that are there with the inflammation they call scar tissue. Next slide, please. And normally muscle regenerates quite well. So if you damage your muscle, if you get punched on the arm and you get a big bruise, you're going to have some muscle fiber damage, but the muscle fiber is going to repair. The same when you have damage caused by FSHD. The DUX4 protein damages the muscle, some muscle fibers die or degenerate, and they are repaired because you have stem cells in your muscle that can repair it. And this is just an example of a piece of muscle. Those two black muscle fibers are newly made muscle fibers that are growing to replace lost fibers. So the muscle stem cells are making new muscle fibers. Next slide, please. But this is not able to sustain muscle in FSHD. So on the right, we have a piece of FSHD muscle that is looking dystrophic, but relatively okay. On the left is an adjacent piece of muscle that looks much worse condition, lots of scarring, lots of fibrosis. Next slide, please. So our aim essentially is to try and make muscle regeneration better in FSHD. We know it occurs. We know that you can replace muscle fibers that are damaged and lost. So we want to understand how you do that. And one important thing to note is that DUX4 has parts that are very similar to something called PAX7. And why this is important is because PAX7 is a very important regulator of muscle regeneration. So PAX7 controls muscle stem cells. And the idea might be that if you've got DUX4 present, you upset the function of PAX7. So that may then affect the ability of the muscle to regenerate. Next slide, please. Muscle is, as I commented, a muscle fiber, lots of muscle nuclei controlling a common cytoplasm of this big cell. And essentially what you have is single cells, myoblasts, that fuse together to make a myotube. And you can see this in culture quite nicely. So below is a panel of cells that are myoblasts, myogenic cells, muscle cells. They proliferate and then they will differentiate. They will fuse together to form muscle fibers. Next slide, please. And if you look at muscle cells from FSHD patients, they tend to make smaller muscle fibers than control. The top ones are from unaffected individuals, big fat muscle fibers, and the bottom ones are from patients, smaller muscle fibers. So if we are essentially trying to look at things that can make the muscle fibers grow bigger, which we think should help augment the muscle regeneration in response to damage. I think we're nearly finished. Next slide, please. Thank you. So we've just been looking at various things. One thing we found here was a dietary supplement called Biocanin A that was quite useful. So we fed the cells that, and it seemed to make the muscle fibers get bigger as they differentiated. Next slide, please. And we're looking at quite a lot at oxidative stress metabolism, how your body makes energy. You need energy for muscles to contract, and you have a whole series of chemical reactions in the body that make this energy in the form of ATP. So we're now examining how you make energy in FSHD and whether DUX4 affects that process, based on a clinical trial from a few years ago that showed that antioxidants had a beneficial effect in some aspects of FSHD. I think it's the last slide now. Thank you. So these are obviously the people that do our work, and we are obviously grateful to our funders. MDUK have funded us consistently over the years. So we're very grateful to them, and obviously we get funding from FSH Society and other learned bodies, and the group that do the work, how they now look through Zoom. Thank you very much.
Wonderful. Thank you so much, Peter. We're going to hear from Dr. Popplewell now, and then we'll have a break for a few questions. And I can see there are one or two coming in already, and then we'll go on to the other talks. So Linda, over to you, please.
Thank you. So hopefully Kat will share my slides.
It'll be there, yeah.
Perfect. Okay, so it's a real joy to be invited to take part in this seminar series, and it's so exciting to be able to contribute, and I really appreciate Kate and MDUK inviting me, so thank you for that. So I'm going to tell you about some of the work we've been doing at Royal Holloway to develop what's called antisense oligonucleotides for FSHD. So thank you to Pete for doing such a fantastic presentation on the genetics and reading of genes. So hopefully it's going to make my job much easier. So on the next slide is just a summary of the pathology of FSHD, but I don't think we need to go through that. And then on the next slide, thank you, Kat, is the genetics recapped again. So I don't need to go through that because Pete did such a fantastic job. But the only thing I will point out is that we have the unlocking of the DUX4 with the decrease in the methylation, but we also need to have this stabilizing element coming into frame, as it were, so it needs to be mutated, and that's what we have in FSHD. So at the end of a gene, we have some elements that allow the stabilization of the RNA that Pete described, and also the processing of the RNA, and that's called the poly-A signal. So it's a stabilizer. So in FSHD, we have the unlocking, but we also have the stabilizer. So what we're doing with the antisenses is trying to block that stabilizer so that the DUX4 RNA is no longer stable and it will get degraded. So antisenses have been used for other muscular dystrophies, but basically they're short DNA sequences, and you can make them specific to the target that you want to hit. So they bind very strongly and hopefully will block the expression that you're after. So on the next slide, so this study started 5 years ago. So we worked with Julie Dimensau, trying to optimize initially antisense oligos to various parts at the end of the gene. So it's not just the poly-A signal, there's also what's called the cleavage site and the downstream elements. So the three elements all work together in the processing of the RNA to allow expression. So we optimized antisenses to the three elements individually and compared them for their effectiveness at knocking down DUX4 expression, and also some of the genes that Pete mentioned that are switched on, that shouldn't normally be switched on in muscle. And we did this in muscle cells like Pete has used. So we treated the cells with the antisenses, and then we looked at the RNA to see what was being expressed. And you can see that when we block the poly-A signal and the cleavage sites, so the two oligos that are highlighted in red in the graphs, they seem to be the most effective, the CS3 and the PAS, they seem to be the most effective at knocking down DUX4 expression. So we went on from that study. So on the next slide, we then designed antisenses that would block both the poly-A and the cleavage site at the same time to see if we could enhance the activity. So again, we used patient cells and we treated them with the different designs that we made, and we compared back to our original best, the CS3, and you can see that on the graphs, what's highlighted, that we're getting much better knockdown of the DUX4 and also the downstream genes on the bottom row of graphs with our PAX S4, that's what we called it. And we decided that that's our best oligo, and we've taken that forward to test in a mouse model. So on the next slide. So development of therapeutics for FSHD has been frustrated by not having a good mouse model to test things in, but in the last couple of years, 2 or 3 models have come through. So after lots of trials, we ended up using the FlexDux mouse, which seemed to work very well in our hands as a model. So so don't worry too much about what's written underneath the graphs, but basically we have the mouse and we treated weekly with the antisense oligo, and we did various assessments throughout the treatment. So it's a month's course of treatment, We looked at various things, but the first thing we looked at was body weight. So you can see on the left-hand graph with the squares, that's the control mouse, and then the blacked out squares, that's the disease mouse. And you can see that the weight drops off very rapidly in the disease mouse, but when we treat with our PAX4 oligo, we do get an improvement in the body weight. So it looks like we're preventing some of the muscle loss. And indeed, when we look at individual muscles, so on the right hand of the slide, there's 3 different muscles. So we weighed them at the end of the experiment, and you can see in the disease mouse, which we're calling the scrambled, there is a drop in weight in all 3 muscles, but the antisense seems to improve or prevents that loss. So we look like we are protecting against muscle wasting. So that's really exciting. We then went on the next slide to look at animal behaviour. So that's obviously important because we want to make sure that the muscles can function, the improvement in the function of the muscle. So here we did 3 assessments. So locomotor behaviour. So basically we put the mice in an activity cage and we monitor their activity over 30 minutes. So we looked at 3, well, lots of measures, but 3 measures are shown in the graph. So we looked at the total time they're active, the distance they travel during that 30 minutes, and the amount of time they're going up on their 2 back legs. And you can see that the scrambled, the diseased mouse, there is a dramatic decrease in their levels of activity. But with the PAX4 treatment, we do see an improvement. It's not completely back to normal, but there is an improvement, which is great. And we also looked at that treadmill exercise activity. So this monitors their resistance to fatigue. So basically we put them on a treadmill and let them run for as long as they can, and we measure the time that they can run for. And again, we did this at 2 different time points, but again, you can see that in the diseased mouse, the black circles, then they get tired much more quickly compared to the control mice. And then with the grey circles, with the PAX4 treatment, there is an improvement back towards the control mice. And then the last measurement we did was called electrophysiology. So for this, we take a single muscle and we contract it, we pull it and see how much force the muscle can generate. So it's a measure of strength. And you can see on the, the left hand of that block of 2 graphs that in the wild type, they can produce very high levels of pull when they contract. Whereas in the diseased mouse, the bottom line in the black squares or circles, they're not as strong. But with our PAX4 treatment, it improves. But when we take into account the actual size of the muscle to make it a fair comparison, so you remember on the slide before that the diseased mouse muscles are smaller, so we have to correct for the size of the muscles. Yeah, so the TA is smaller, but that when we correct for the size of the muscles, that the PAX4 treatment almost gets back to how the control mice look. So it looks like our PAX4 is working very well in terms of improving the muscle function. So that's really exciting. And on the next slide, we checked what our antisense is actually doing, because obviously we're trying to target DUX4. So we needed to check the DUX4 levels of expression. So on the top line of graphs, we looked at DUX4 and then 2 of the genes that are normally switched on. So you can see in the control mice that it's not normally expressed or at very low levels, the 3 genes. But then obviously in disease, we're getting DUX4 expression, and then the 2 downstream genes are switched on. But with PAX4 antisense treatment, we're getting, maybe towards 50% reduction in the expression of those genes. So it's not complete inhibition, so there is still room for improvement, but we are obviously hitting our target, and we're seeing an effect on the downstream genes, which are what cause the toxicity in the muscle. We also looked at some of the genes involved in the phenotype of the disease. So we looked at genes involved in inflammation that Pete mentioned. So you get inflammation in the muscle, and our PAX4 treatment does reduce expression of IL-10 and TNF-alpha. Next slide, please. And then we looked at how the muscle looks. So as Pete described, we're sectioning the muscle and then we're staining and looking at expression of various proteins. So this is again looking down the length of the muscle. So on the top graph and the images, we're looking at the size of the fibres. So as Pete said, the fibres get smaller with disease, and you can see that our PAX4 treatment sort of makes the muscles bigger, which means that we're preventing the muscular wasting, which is really nice and fits in with the absolute weight of the muscles that we had on the previous slides. We also looked at the degeneration, regeneration, which is obviously what leads to the muscular atrophy. And again, we're preventing the expression of the genes that are normally involved in that process. And we're also preventing the fibrosis, so the scarring in the muscles. So we've stained here for collagen, which is one of the fibrotic proteins, and we're getting a decrease in the amount of fibrosis. So it seems to be targeting the three main pathways that are involved in the pathology of FSHD muscle. So it looks very positive. So next slide, I think, just summarizes everything. So it looks like using Antisense, we can block the expression of DUX4 to a degree, and that does seem to improve the muscle function and the activity of the mouse model. And it seems to prevent the pathology that's seen in the muscle sections. So it looks like it could have promise, and there are a number of companies working to optimize the delivery. So I think, you know, we're getting 50% reduction in DUX4 expression, so there is room for improvement, and I think part of that will be optimizing the uptake and the delivery and getting the dosing right. So there is hope, and you know, there is still more work to be done. So all of this work was performed by Nok-Lu Nguyen, who's a fantastic scientist, and I'm so lucky to have her in the lab. And we've just very recently published this work, and it's an important paper because it was the first to describe treatment of a whole mouse with an antisense. So there have been previous papers where they're just doing local, you know, single muscle injection, which, you know, for FSHD, we need to treat more than one muscle. So we needed to show in, you know, a whole body treatment that we can see an improvement in the muscle function and pathology. So it's looking really nice, and we're really excited to have been able to have done this work. So I'd also like to thank on the next slide everyone else in the lab at Royal Holloway, Julie for collaborating with us initially on the first study, Vincent Mooley, who provided us with the patient cells, and also Peter Jones for his FlexDux mouse and all the advice he's given us working up that model and then doing the in vivo study. And obviously MDUK, none of this would have been possible without them. So I thank them for all of their support over the last few years with this study. And thank you for allowing me to share.
Thank you very much, Linda. Before we go on to the next talks, which are much more sort of clinical in focus, there was a question that has come through. Really kind of for you, Peter, and Linda, if we can touch on it sort of quite quickly. Peter, you spoke about the number of repeats of the DUX4, and then, you know, fewer of those repeats leads to FSHD. Is the number of repeats that you're missing, does that correlate with the severity of the condition, or is that not been found yet?
No, it does seem to be, so that if you have one or two repeats, that can be associated more with the congenital onset, so the onset from a very young age. If you've got 8, 9 repeats, 10, you're borderline, it tends to be later onset with less severity, and kind of, so there is a correlation. It's not an absolute correlation. There are other confounding factors, but there does seem to be an association.
Okay, thank you.
And that's FSHD1. and FSHD2, you have more units, but you have a mutation in a different gene that is important for methylating at D4Z4. So again, the fewer you have of the repeats and this second mutation is more severe. So it makes FSHD1 worse, as well as making FSHD2 worse, the number of repeats.
Right, thank you. Thank you, that's very helpful. And does the poly A tail, all of those A repeats that you were talking about, Linda, does the length of that, does that have an impact on the severity?
No, so the poly A and all of the other bits that are at the end of the gene, they're fairly conserved between genes, so they don't vary so much. Obviously there's different sequences around, but that doesn't get mutated. It's the fact that there's another one that comes that is mutated in DUX4 that allows it to be expressed. So it's, you know, there are a number, usually a number of the poly-A signals on a gene, so they can all be active, but normally the one that doesn't work, and obviously it's silenced through the methylation, keeps it not being stable. But obviously in FSHD you do get that single mutation that allows it to act as a poly-A stabilizer. Right. But it doesn't change length, it's just a single base, normally a single base change.
Right, I see, thank you. So it's the DUX4 and the numbers of those that really are the critical thing. Wonderful, thank you so much. Let's move on to the next set of talks, and we're going to hear now from Dr. James Lilliker, who's going to talk a little bit about the kind of clinical research that's happening in FSHD. So over to you, James. Are you muted? James, I can't hear you.
Hello, can you hear me now?
Yes.
Sorry about that, the best laid plans, and then I just turned to my other screen and managed to disconnect my monitor, which is rather unfortunate. So hopefully you can see and hear me now. Do let me know if that's not the case. Yeah. Right, next slide, please. So just, yes, I've got no disclosures. Next slide, please. Just a little bit about me. I mean, I'm not a sort of FSHD expert or an expert in FSHD research, but I co-direct the Muscle Diseases Unit at Salford Royal with Mark Roberts. I see lots of patients with neuromuscular disease at the sort of coal face, if you like. I developed and now lead the service for patients with spinal muscular atrophy, and we administer nusinersen and other treatments to them, and we have around 50 such patients now in the Northwest. And I've got quite a bit of experience running commercial clinical trials, but also setting up a more homegrown research program focused on inflammatory muscle diseases, and I've recently completed a PhD on that topic. Next slide, please. What I wanted to talk about today, just briefly, is not necessarily what's going on in clinical research in FSHD, but why research is important, and perhaps some controversies around clinical research, and a competition that research has with other aspects of care. So clinical research is really important. It's been shown that hospitals where there's a lot of clinical research activity have improved outcomes and better care that's delivered to patients. We know that by doing research, we can find ways to prevent the development of disease, prevent the recurrence of disease, and hopefully reduce the number of people becoming ill. We know now, and in patients that I see on a daily basis, that people living with neuromuscular diseases face significant and ongoing unmet needs, high levels of disability, missing out on life chances, struggling with their education, struggling with their employment and travel. And really, it's only by doing research, or at least research is an important aspect of this, that these unmet needs can be resolved, and we can improve survival and improve quality of life and reduce that level of disability. Finally, and I do put it last deliberately because this is the most difficult, research does provide the opportunity to find cures for disease, and hopefully increase the number of people who live their life in good health. But as we all know, this does take a very long time. Next slide, please. So I mean, are there any downsides? It sounds great. Why don't we, why are we all doing research all the time? Well, I think unfortunately there are some downsides which we have to be aware of. Clinical research is not always equitable. People living close to a neuromuscular centre may find it easy to take part. They may be more likely to be invited to take part. So there's not always fairness across the country, even in quite a compact country like the UK. You do see some of this perhaps unfairness in who's taking part in clinical trials. People that have the financial stability to take time off work and travel repeatedly to the specialist centre might also be more likely to take part, and I think that's an important issue. And then the other thing is that the clinical trials often have very strict inclusion-exclusion criteria, and people will miss out because of this. Perhaps they're a few years older than the upper age limit of the trial. Perhaps they incidentally found to be vitamin D deficient, just very arbitrary rules which mean that they can't take part. There's also risk to being in a clinical trial. Obviously, we're trialing new treatments. People sometimes say that they feel a bit like a guinea pig. Sometimes people say, this isn't really benefiting me, is it? It's more just to do with helping people with my condition at large. And I think these are important issues that that people have to be aware of when they're taking part in clinical trials. Now, I mean, the important thing here to remember as well is that clinical trials are very complicated to set up. They're very expensive to perform. They require massive investments in time and infrastructure. And often clinical research is funded through private organizations, pharmaceutical companies and others. And at the end of the day, they're expecting a return on their investment. And because of this, the focus of research programs could be seen as being financially driven and not necessarily patient-led. So sometimes we might not be focusing on addressing those real unmet needs I described before of people living with neuromuscular diseases. The big question, therefore, is could our time be spent more effectively elsewhere? Next slide, please. The other problem is that most research findings never make it into the clinic. And really, why is this? Maybe the study had negative outcomes, and that's important in itself. It's important to avoid treatments that don't work. A lot of the time, though, in neuromuscular disease, it's because the trial was very small, the results were inconclusive, or the results couldn't be replicated. And essentially, that trial in isolation, people might look back and say, well, what was the point in that? It was too small, really, to give us the answer we needed. Sometimes the reasons are even more complex. Even if a study finds a drug that works, this drug is likely to be very, very expensive. The drug might be very complicated to administer, require significant changes and investment in staff and infrastructure. So thinking about genetic therapies or intrathecal therapies, for example. And finally, sometimes clinical trials show a drug works in a statistically significant sense. So there is a difference between those that had it and those that didn't. But the actual size of effect can be very small. And then when you translate that into clinical practice, really, people ask whether it's worth the investment. And that is the elephant in the room. We have to contrast this with the fact that healthcare funding is limited. In providing one treatment, does it mean that another cannot be afforded? And should the priority be on funding these new drugs, or perhaps investing in the overall healthcare system, including medical, nursing, therapy, support, and social services? Next slide, please. So should that focus be elsewhere? And it is important to recognize this is something I say to patients every day. Just because your disease is incurable doesn't mean that nothing can be done. Thinking about FSHD, have we got a specialist nurse? Have we got some therapists? Do you have an exercise program? Have you got your ankle foot orthoses sorted? Have you seen a shoulder surgeon if it's relevant? What's support like in the community? Wheelchair services, why are you waiting a year for your wheelchair? Is your pain under control? Is your fatigue managed? Do you have that psychological support? And really, I think if we got these things right, this could have a huge and transformative effect on the lives of people living with neuromuscular diseases. Next slide, please. But of course, these things aren't mutually exclusive. We need to do both. We need to do research, but we need to get the basics right too. Some people might say to me, oh, this is just a false choice. It's never sort of one thing or the other. But I would argue this is a choice that we have to make as a society and within the NHS. Resources are limited. There's a limited number of neurologists. Their job plans are already full. There's limited numbers of nurses. There's limited space. There's limited expertise. There's limited funding. We do have to make choices. But it's important to remember that there's also huge scope for research to actually save money for the NHS. We can avoid drugs that don't work, and we can find simple, cheap things that do work. So, maybe non-drug treatments, for example. And, coming to the end, I would say that any new treatments are likely to have a much bigger and more sustained effect on their quality of life if they're accompanied by those overall improvements with the care delivered, including access to therapists and an exercise program, for example. So I think it's critical that research continues. It gives us a hope for a cure for FSHD and other neuromuscular diseases. But when we're banging the drum for more research and new treatments, I think it's important that we don't forget the basics. They're just as important. Next slide, please. Thank you very much.
Thank you very much, James. That's a very great overview of clinical research and its importance. We're going to hear now from Dr. Bugiardini. I believe we have a video of Enrico. So, Kat, have you got that all lined up and ready to play?
Yes, just 1 second and I'll bring it up.
Wonderful, thank you.
Hello, my name is Enrico Bugiardini. I'm a clinical research associate at UCL in London. And first of all, I would like to do a very quick background on the place where I work. I work at the Queen Square Centre for Neuromuscular Diseases. That is one of the largest centers in UK. We see over 6,000 adults per year, and we have more than 20 specialist services. The center is collected by Professor Anna and Professor Relly. And as you can see, there are several services that are grouped depending on the condition under study. Within the muscle services, we have also established an FSHD dedicated clinic that is a multidisciplinary clinic in the sense that there is the input of multiple specialists, including a specialist physiotherapist and a clinical nurse specialist. The core missions of the centers are to provide the highest quality patient-centered clinical care, to provide excellent education and training, and to undertake world-class research in order to understand disease mechanisms and to develop treatments for patients with neuromuscular diseases. In terms of clinical research at Queen Square related to FSHD, there are 4 key areas that we are trying to develop. The first is to improve diagnosis, because the current diagnostic approach in FSHD is limited. And there are still a lot of people that, despite having common symptoms typical of FSHD, we are not able to confirm the genetic diagnosis. And giving the upcoming clinical trials and potential treatments is paramount to be able to provide an accurate and rapid diagnosis to those cases. It's also important to improve quality of life, which is another area of research, because the upcoming clinical trials are aiming to stop disease progression, but we still need to manage FSHD-related symptoms, and not just related to mobility issues, but also pain and fatigue. Biomarker discovery is another area of research. Biomarker is an objective measurable indicator of severity or presence of the disease, and biomarkers are so essential for clinical trial development. At the moment, there aren't optimal biomarkers in FSHD. A clinical trial preparedness, and in this context, I will be speaking in this lecture about 2 networks that we are joining, the FSHD Clinical Trial Research Network and the European FSHD Trial Network. In terms of the FSHD Clinical Trial Research Network, it was established in US and is led by Dr. Rabita Will in Rochester and Dr. Jeff Statland in Kansas. During the years, the network has expanded and involved many centers in US, but also centers in Europe and in UK. The main aim of the network is to close the gaps in trial readiness, because they soon realized that a major obstacle in clinical trial development was the lack of an existing clinical trial network with common standard operating procedures. So the FSHD CTRN overcomes this challenge by creating a streamlined system for regulatory oversight, regulatory approval and ethical oversight, by developing standards for what data is collected and how it is collected, by creating a network of clinical evaluators and study coordinators, and by validating new outcome measures. So the FSHD CTRN believe that with this structure, we will be able to be ready for efficient conduct of clinical trials in FSHD. And so we will also be able to perform large-scale studies and also to attract pharma interest and to improve patient care and outcomes. One of the best examples of the FSHD CTRN is the RESOLVE study, that is an acronym for Clinical Trial Readiness to Solve Barriers to Drug Development in FSHD. This is one of the largest studies in FSHD that aim to recruit more than 200 participants. The overall aim is to hasten drug development for FSHD. And there are several gaps that it aims to resolve. The first and most important is on outcome measure. An outcome measure is a measure used to assess the effect of a treatment. But at the moment, there aren't optimal outcome measure for clinical trial in FSHD. So in the reserve study, they try to validate and assess in multiple centers 2 new clinical outcome assessments. Another challenge is variability of progression in FSHD, and the variability of progression can have a negative impact in clinical trial results and development. Given the size of the study, the investigators believe that they might be able to then determine subgroup of patients with homogeneous progression, so to try to stratify the participants for future clinical trials. And then biomarkers, because there are a big amount of data and sample collected, those samples may help the investigator to identify new biomarkers. These are the 2 main outcome assessment of the study, and I thought it was useful just to show them to you, to have a practical example of what consists the study. So the FSHD Composite outcome measure is made by a multiple assessments designed to fully capture changes in disease progression, specifically for FSHD, because we do have several assessments for muscular dystrophy for clinical trials, but we would need something tailored to FSHD. And as you can see, there are several regions that are assessed with multiple tests, including leg function, shoulder function, trunk function, hand function, and balance. So we are sure that we are able to monitor any changes in any of these functions. The other outcome measure is the electric impedance myography. This is a new tool for quantifying the muscle structure. It uses a subthreshold electrical current that flows through the muscle and is able to detect the ratio between lean muscle and fat tissue. So it might be able to identify early muscle changes and to monitor them during a trial. I would like also to speak about the other network that were involved. The other network was started by FSHD Europe. FSHD Europe is an organization supported by several FSHD related charities, including MDUK. FSHD is a doorstep of clinical trials, and FSHD Europe has realized that there are differences between European country and US, and in terms of guidelines for clinical trial, pharma regulation participation, and healthcare provision. So they thought it might be useful to have a network, particularly focusing on European country and European regulation. So the main aim of the FSHD European Trial Network is to connect the European clinicians involved in FSHD, is to facilitate the setting up of future clinical trials, and to make future treatments more accessible. There are other specific aims that I just list some of them, just to give you an idea of the scope of the network. They also want to harmonize between the European centers criteria for clinical and genetic diagnosis, harmonize the registry, the protocol, the treatment, and obviously they would like to engage with pharma and EMA for a Europe-wide collaboration. So the milestones done so far are reported here. So in early 2021, the FSHD Europe nominated the executive committee. They then contacted FSHD experts in several countries to create a core group. The core group met in April and May 2021 in a virtual workshop to discuss clinical trial preparedness in general in FSHD and more specifically in Europe. And they agreed a strategy in the creation of 4 working groups that are working on clinical and genetic diagnosis, outcome measure, biomarkers, and imaging outcome measure. These are 4 core features of clinical trial preparedness. So I'm very pleased that UCL and UK are connected with both networks, the FSHD CTRN and FSHD Europe. The benefit of being part of these networks are numerous, because we can be involved in large-scale research projects such as RESOLVE. We are updated with trial readiness for upcoming clinical studies. We can enhance UK-based research using the infrastructure of the network, and we can facilitate the training of the next generation of FSHD clinical researchers in UK. So I think it's a very exciting, promising moment for FSHD research, and we won't be in UK, we won't be up ready to collaborate and to support the international efforts to finally find a treatment for FSHD. Thank you.
Thank you so much for that talk, Enrico. If we could play the video from Ben Porter of the registry, and then we'll answer some questions after that.
So, what is the UK FSHD Patient Registry? So, first and foremost, what is a patient registry? So, it is a research database that contains information about individuals who are affected by a potential condition or disease or genetic mutation. In rare diseases, they actually play a really important role in helping develop new therapies. The UK FSHD Patient Registry is a registry that collects clinical and genetic information about both FSHD type 1 and type 2 in the UK. Patients complete a questionnaire that's on a secure online portal. This is then combined with some doctor-verified clinical and genetic details. You tend to select your doctor during the registration process, and this will often be a neurologist. As you can see on the right-hand side here, this is part of the homepage and where you can register online on the UK FSHD Registry website. So why join the UK FSHD Patient Registry? So, well, the registry itself helps identify participants for potential clinical trials and research studies. The registry helps in the development and standards of care and can help improve the care that people actually receive. The registry can support specific research questions for doctors and scientists, which helps better understanding of specific areas associated with FSHD. It can also contribute to the natural history of the condition, so it'll help us understand more about how the condition and disease course looks like over time and how it might be altered by treatment in the future. The registry also helps provide a link to the research community, enabling people to receive information directly from the registry. So things like newsletters, research updates, or potential updates on standards of care when they happen. So how do you join the UK FSHD Patient Registry? So as I mentioned at the start, the registry can be accessed online via the registry website. There's a link provided. Patients or caregivers can actually initiate registration. You provide some basic demographic information, so like name, date of birth, some contact details. Informed consent that is then required before completing the registry questionnaires. And again, as I mentioned at the start, you'll also be asked to select your doctor from a list of doctors who are affiliated with the registry who give permission to be on there. If your doctor isn't on there, then I'd recommend that you contact the registry curator, myself, and we can try and contact your doctor as well to try and get them on the registry. And then once you provide that information, it allows us managing the registry to potentially work with academics, researchers, patient organisations, and potential pharma companies who may be running clinical trials. It allows us to then share de-identified data, so that's information that does not identify you, often known as aggregate data. As long as we have the approval of the steering committee, we can potentially share that information with those groups to, for the purposes of clinical trials or other research studies or data analysis, which brings us nicely to this section. So the use of the FSHD registry in research. So as I mentioned, we can support a wide range of different groups. At present, we have actually supported about 30 different inquiries since 2013. So it's not limited to promoting clinical trials and research studies and supporting their recruitment, promoting clinical and academic surveys to support various research projects, and also providing de-identified anonymous patient data to researchers to support data analysis. So examples of the actual use of the registry in research. So in I think it was in 2019 or last year, we supported recruitment to the FSHD Society's Voice of the Patient Forum surveys. So the registry circulated 5 surveys that aimed to support the patient-informed drug development to over 900 patients in the UK. Overall, we had nearly 100 people actually complete all those surveys, which made up about 15% of the overall survey recruitment. So most of that survey recruitment was done in the US, but there's various other countries who were able to share those surveys as well to reach wider FSHD communities. In 2019, we also supported recruitment to a patient-focused survey which assessed the proposed clinical study design in patients with FSHD. So that was with Fulcrum Therapeutics. And we were very fortunate that the registry was able to recruit 41% of all the patients involved. And the company did assure me that the feedback from patients on those surveys was incredibly important to them designing their clinical trial protocols. And then finally, an example about sharing de-identified data. So the registry shared some data with a researcher from King's College London about factors that affected FSHD symptoms. So data from 730 patients on the registry was analysed, detailing a whole wide range of variables, which included age, sex, symptom onset, D4, Z4 length. And this has actually resulted in a publication in a scientific journal. So you can find that at the link there. Alternatively, you can also find a list of publications that are associated with the registry or directly involving the registry on the new section on the registry website called Publications. So we detail all publications that we've been involved in since 2013, and this is regularly updated. So if you click on the title of the publication, that should take you directly to that publication itself. We've also updated another section on the registry called Project Supported, which again makes it really clear and transparent all the projects that we have supported. Of course, if there was any companies or researchers who wanted things to remain confidential, we wouldn't put it there, but thus far we've been able to document all 30 or so inquiries that we've been involved in, which I think is really, really good for everyone to see. And where I can, I've also reached out to those researchers to understand how the registry itself has supported recruitment to those studies. So, you know, whether or not it was 50% of the recruitment total came from the registry, for example. I've also, as some people will be aware, been updating the FSHD drug development pipeline that I've been including on the registry website since mid-2019. Again, this is an ongoing process, but it shows everyone what's in development to treat FSHD. So, you know, whilst we know there's a few clinical trials, there's lots being done in what's known as the preclinical stage of development. So, you know, being studied in animal models such as rats. So again, I've documented this all in this infographic and have provided links under the section called Source of Information in case anybody is interested in learning more about these developments. So to conclude, patient registries themselves are a versatile research resource. Hopefully from what I've told you there, you can see that patient registries themselves can help facilitate more than just clinical trial and research study recruitment. They're widely beneficial to both patient, researcher and drug developer. If you have the opportunity in registering with one, I would highly recommend this. Obviously I'm biased because I run the registry. If you're interested in finding out more about new studies and clinical trials, they will often appear on clinical trial registers such as clinicaltrials.gov, which is widely used in the US and used quite a lot internationally, or the clinicaltrialsregister.eu, which is a European clinical trials register. By being involved in the patient registry, there is potential to be notified and be involved in a wide variety of research that will not appear on those clinical trial registers, however. So I'd like to thank Dr. Chiara Marini-Bertolo, who is the registry principal investigator. I'd like to thank all of the researchers, doctors, companies, patient organisations, patients and caregivers who've helped support the registry over the past 9 years. And a shout out to the steering committee who have helped govern the registry as well. If you have any questions, please do not hesitate to contact the registry at the email address provided there. And thank you for your time, everyone. Take care now. Bye-bye.
Thank you, Ben, in your absence, and Lindsay, there are some questions for you coming up. So we've had lots and lots of questions. I've got quite a few to get through. I'm mindful of time, so if you can be sort of thorough and yet quick in your responses. I know that's a kind of somewhat tautologist thing to say, but if we could perhaps start with some of the going back to some of the work that Linda and Peter were talking about. Linda, your studies are really exciting, but you know, these were studies in mice. Do you have an idea of how long it might take before things could be in human trials, and even then into the NHS? And that's a big question, but
yeah, that is a big question. So FSHD Society had a conference a couple of weeks ago. I mean, we're not the only ones trying to develop antisense oligos for FSHD, but there are a number of companies that are now rapidly developing towards clinical trials. So like I said, they're trying to enhance the delivery. So I've just looked back at my notes, and they're all sort of talking about last quarter 2021 going into clinic, and also 2022. So I think they are coming, and the graph that the last speaker just showed, you know, with the sort of pan of trials and where they are, I think there's a number of companies that are almost ready to go into clinic, to a clinical trial.
When you say clinical, it's clinical trial rather than in the clinic. Yeah.
So there's still a long way to go before it would be available through the NHS. But they are all US based companies. So I imagine the trials will be focused over there, but there's no reason why, you know, patients can't get involved in the UK through the registry. But yeah, so it is moving, but obviously it all takes time. So
Sure.
to be able to say when it might be available, I don't know.
And similarly, I guess, Peter, some of the stuff that you're talking about, especially related to the PAC7, you know, you're looking in cells, you're looking in mice. Is there any sort of, I suppose, line to getting towards a clinical trial, sort of line of sight there?
First, on the anti-oligonucleotides, Duchenne has already had clinical trials, so at least it's a kind of proven technology, and it's got through to phase 3 clinical trial there, so that's encouraging. Our stuff is more basic, so we're trying to understand the mechanisms, and by understanding the mechanisms that go wrong, it points out potential interventions. So as I think James was saying, was interesting, it's not potentially always a cure, but right now, if you could just find something that could help on a daily basis that's over the counter or a dietary supplement, that's quite useful. So that's kind of more where we're going with the energy generation type.
Right. And you mentioned a supplement. Is that something that people can access and they could reach now?
I mean, that is very basic preliminary data, so that I'd be more caveated. But the fact that there was a clinical trial with antioxidants that, you know, is published, and it was a 17-week trial, and people reported improved strength. So I think that is encouraging. I mean, you don't want to go over the top with antioxidants, but they were just standard vitamin C, selenium, zinc, over-the-counter stuff. So the research is out there. The references in the my talk, so people can read it and see what they think, basically.
Brilliant, brilliant. And talking of clinical trials, I mean, many of the people who are viewing this session will have heard of the Fulcrum trial that published some data last week. Enrico, turning to you with your expertise around the sort of the clinical trial network, the research network, and what have you, do you sort of get a feeling for when that might come to the NHS, might come to the UK? I mean, I realise, again, Fulcrum is a US-based company. They are focused in the US at the moment, but have you got anything to say on that?
Well, I think it's difficult to say, because first the Fulcrum has to discuss with the FDA, I mean, to see if they can get an accelerated approval or not. So I think once it's sorted there, if they need further data, or if actually they can get an approval, then there can be another discussion within the UK and MHRA and I mean, in our regulatory body. So it's obviously they are different from the one from US. I think the positive things of being in the network is that we might be involved potentially in other studies in future with other molecule, or if they need to extend, I don't know, hopefully there might be more chances to be involved, but to be actually available directly in the NHS is very difficult to say. To be honest, it's very preliminary, because The data were very positive, but I think a lot of people read about the fulcrum result, but primary endpoint actually about the DAX4 inhibition was not satisfied, but they did see some positive effect on clinical measure, but we don't know if those are enough for the FDH and our regulatory body to be approved quickly.
The main issue is that the drug is quite wide ranging. It's not specifically, it's meant to affect DUX4, but it also affects lots of other cells because it's a pathway that's used by many cells. So right now, it seems to be there's an effect, but no idea of how the effect is being caused, because the primary measure, which was meant to be DUX4 target genes going down, wasn't met.
Thank you.
Yeah, I think the issue there was also about that that we were not sure that the way of assessing DUX4 expression was the best one, because it's very difficult to detect it, and especially when you do a trial in human being and you try to assess muscle samples with a needle biopsy. I mean, you may sample a part of the muscle that is not much informative. Yeah. And that's what they are saying now. So they were very pleased that actually the clinical outcome measure worked. But it would be nice now to really find a way to measure a definitive effect on DUX4 expression. That's why also all the studies, research on biomarkers is very important. And mainly, I have to say that I just believe it's a positive result, but it should be more a push to do more and to find also other treatment and more research, basic research on biomarkers. It shouldn't be the rival point. But it's positive because it showed some positive results. So I guess other pharma and other academic centers, they are even more interested because they see that potentially we could do something, but it shouldn't be the rival point, to be honest.
Sure. Fantastic. And just again to sort of update everyone, Rob and I will be speaking to Fulcrum Therapeutics next week to find out what their plans are for actually coming to the UK, but and when we can, we'll be able to share some information, but it will be a long journey to get to the UK because of the things that you mentioned. Talking of clinical trials, Lindsay, people can register with on the registry. That sounds such a strange thing to say. Yeah. They can share their details with the registry, and that's a way potentially to find out about clinical trials. But there have been a couple of really interesting questions about who can access the information, personal, very private information about people, who has access to that, and also, you're sharing that the data is shared potentially with pharmaceutical companies. Is the data being sold at all?
That's a very, very topical question. To take the first part then, who has access? Potentially, the data is available to all people, but as Ben mentioned in his presentation, it is only de-identified and anonymized data is available. What we encourage is if you would like to find out information from the registry, to put in an inquiry to the registry, and that is considered by the steering committee, and they govern the operation of the registry and will review any applications for data. And it's only upon their consideration and approval will any data be provided. Often we've had inquiries from patient organisations, from individual researchers, as well as, of course, healthcare providers, but also pharmaceutical companies. But again, stipulating that any data that is released is de-identified and anonymised. In terms of selling data, in the past, no, we have not recuperated any financial support. We're beginning to change our model, but purely so that we are able to sustain the registries, because we exist very much on the goodwill of patient organisations and their support to enable us to continue to operate. And so in a few instances where inquiries are made, we ask for a little bit of support to sustain the continuation of the registry.
Fantastic, thank you. couple of questions again, going back to the DUX4 issue. Do people within the same family have the same number of repeats? You know, if they've got FSHD, is it seen from one person to the next? Maybe Pete, you know the answer to that one.
Yeah, so it's you can inherit it. It's spontaneous as well. So sometimes it just happens, and there's no track record of it. But yes, if you have the chance to pass on, I mean, it depends on circumstances, because obviously you will pass on one copy of the 2 copies you have. So it depends if it's the you know, the contracted copy or the non-contracted, and then your partner, depends what they have. If they have sort of 2 healthy legs, then you can do the kind of maths to work out the chances. It's 1 in 4 and things like that. So it depends on what the parents have. But yes, it can be passed on, and I don't think there's a lot of evidence of anticipation. So it's generally passed on as a kind of stable number. The number you have will be passed on.
I see. Thank you. I just spotted a question that has come in about potentially going overseas for stem cell therapies, and particularly for FSHD. Enrico, I wonder whether you can comment on stem cell therapy for FSHD and indeed any other muscular dystrophy.
Well, I mean, I think it's a bit complex topic, because obviously it's something that researchers try to explore and develop, but we don't have yet much practical evidence that it works. Obviously, it's a good path to follow, but I believe, as I said before, that now that we are all part of networks with the US and Europe, and especially those 2 networks are the lead of FSHD research. So if there will be something on stem cell specifically for FSHD, we will be all informed if it works or not works, and the facility or academic centers that can provide that. I always a little bit concerned about, I'm not sure which study we're speaking about, but just checking random centers and try to access therapy if we don't have enough evidence that they work. And that's why we did all this effort to join the international groups, so we can be updated. But it doesn't mean I can be 100% sure that if there is a study will be involved, because it depends from many factors, especially funding, but at least we are aware of what's happening and what are the best trial and treatment available for FSHD people. Yeah.
I think that there are some clinics who in certain countries who may be offering stem cell therapy now without it being actually proven, and that there's that clinical evidence that supports it. So,
yeah, I always, I mean, as a scientist and clinician, I always be concerned about doing treatment that are not approved, also because we don't know also about
the most encouraging stem cell angle is on OPMD, which is another muscular dystrophy, where they've taken some muscle stem cells from the leg and put them into very small muscles in the throat, because OPMD patients have problems swallowing. So that's quite a small trial, but that seems to be encouraging. But they're very, very small muscles. So I think this is probably the issue. With FSHD, you have quite large muscles and quite widespread. So for a very, very localized, very small muscle, that possibly it's looking encouraging. It's still obviously being trialed, but that seems to be about the best example so far, from what I know.
Fantastic. Now, I am very conscious of the fact that we have had lots of questions and we haven't had the chance to answer them all. Many of them we have, but I'm also mindful of the time, and we have promised everyone a very short comfort break. So we will wrap up the research section now. We'll try and respond to some of the questions that are still being asked in the Q&A section. We'll try and answer those online. If we take a 5-minute break now and come back, now by my clock that says 11:58, which is quite specific, but I don't want us to take it too long. We're running a little behind, bad chairing, sorry. And then we'll listen to the session and take part in the session on living with FSHD. So we'll take a 5-minute break. Thank you so much to all of our research speakers and for answering all those questions. It's been truly fascinating, really interesting and informative. So thank you very much for your time. And please, if you'd like to, do stay with us for the Living With session as well. And we will be back in 5 minutes' time. Thank you.
Thank you.
Hello, everybody, and welcome back to the session. Could I just ask any panelists when they're back to come on camera so I can keep track of when we can resume? Thank you. Oh, thanks, Kate. Very good. Hey, Nick, thanks for joining us.
No problem.
I am here, Rob, but I probably have to scoot off quite soon.
No problems. Thanks, James. So I think for this session, it's just Liz, I think, to come back on camera. So we'll just give Liz a couple of extra minutes, a couple of extra seconds. Liz, if you're with us, could you just come on camera? Otherwise, I'll kick off and start with Joe and Nick. Right, I'm conscious of time, so I'll start us off in this next session. So we've got about 30 minutes left in total, about 25 minutes to take a range of questions on living with FSHD, and then we'll be hearing from Raj Badiani for the final 5 minutes. So I'll introduce Nick and Joe first, and then I've been keeping track of some of the questions related to physio. So what I'll try and do is invite you to make a few remarks, but also feed in some of the questions as we go. By way of introduction, Nick Emery is a neuromuscular clinical specialist physiotherapist at the Robert Jones and Agnes Hunt Orthopaedic Hospital in Oswestry. I have been lobbying for that name to be shortened ever since we started these series. Nick qualified as a physio in 2005 after being a firefighter in London for 18 years, and he spent his entire physio career in neuromuscular disease, and works as part of the multidisciplinary team at the centre there. And Joe Reffin is the clinical lead physiotherapist at King's College Hospital, and has specialised in neuromuscular conditions for the last 15 years, and was involved in establishing the long-term physiotherapy-led follow-up clinic at King's. If I could ask Nick and Joe in turn, perhaps just if you could just say a few words about the importance of physio in managing FSHD and why it's such an important part of the team. And Nick, in particular, if I come to you first, there was a mention of the multidisciplinary team. So how do you work within part of that kind of wider team supporting people living with FSHD?
Well, as a centre that deals both with paediatric and adult, we just see patients right the way through. So I think our youngest patient with FSHD is 16 and our oldest is well into their 70s. So as we know, it's probably been discussed that it presents in a wide range of problems. So it's highlighting and having experience on identifying problems before they start, so you can take action so that you can promote as much function as possible. So part of that MDT approach would be that we have obviously 2 consultants, but we also have a psychologist, we have clinical nurse specialists, and we also have access to surgeons who will do various surgeries for improving shoulder function, and also a fantastic orthotics department. So we can call on lots of different specialities, but it's also having that knowledge within physiotherapy that we can prescribe various interventions, whether that be exercise or home adaptations, and combining that with the team so that interventions can be put in place, trying to mitigate any future problems.
Thanks, Nick. Now, you've actually opened the door to one of the questions that's coming already. So I'll ask you a question before I come to Joe, which was around, you've used that word mitigation of problems. And one of the questions was that someone has a diagnosis of FSHD, but so far isn't symptomatic. So can physio sort of delay the onset of symptoms and can mitigate against further damage by sort of strengthening muscle?
I'll give you a horrible answer. It's yes, no, and maybe, because the evidence would suggest that we can help to I wouldn't say strengthen, but probably prolong the ability of the muscle to maintain some strength over a period of time. But the critical point in that is how much, how often, and how intense do you prescribe exercise? I know that one of the questions is about, can you damage muscle through doing exercise? Yes, anybody can damage muscle through doing exercise. And certainly if you've got a muscle condition, you will damage muscle sooner. If they haven't got symptoms, then it's important that they stay as fit and healthy as they possibly can. But mindful of overdoing stuff. And it's very tempting to think I can just go down the gym and I'll just exercise and I'll get stronger. No, you probably won't. And there is a possibility you will damage muscle and that will not then repair and so it will make your condition worse over time rather than better.
Great, thanks Nick.
Trials using exercise. I came in a bit late. I don't know if you've mentioned those, but there's a couple of clinical trials where they said that moderate exercise was useful, but again, as you comment, carefully controlled.
Yeah, I mean, John Vissing's done some work on it, and there was Chris Morse up in Manchester who's done some work, and Chris Morse's trial was quite evident that it was only a very limited number of patients, but his work actually evidenced that doing some lower limb exercise actually improved gait and actually improved what we say is ground reaction vectors. So looking at where the forces go between various points. So there is some evidence that it does do, and light to moderate exercise is probably beneficial.
I think it's fair to say, and I hope you'd agree with me, Nick, that exercise can, in the main, is beneficial for patients, and the risks of causing harm is relatively low. And I think that came out in the recent Cochrane review. They weren't really able to make many recommendations about what type of exercise you should do or how you should do it and how often and things like that. But they were able to say from the evidence available that exercise in general is pretty safe. Whilst there are risks, they're reasonably small.
I'd like.
Yeah, sorry, Jo, go ahead.
No, sorry. Thank you, Nick. And thank you, James, very much for your comments. I think I'd just like to echo what you've both already said, in that I think the physiotherapy role, as Nick has alluded to, is a very broad and very holistic role. And the title of this session, The Living Well with FSHD, I think can be really well facilitated by physiotherapists, both in the clinic and in the community. And I think I feel very passionate about patients remaining active in a way that works best for them, in a way that they enjoy, and in a way they can sustain over the longer term. And whether that is, relatively light aerobic activities such as walking, an exercise bike, or electric bike, water-based exercises, a Pilates class, or yoga. I mean, there are many different activities that patients can be involved with without risking harm. And I think the healthy living, and then what carries on from that in terms of managing your body weight, managing your mental health, your mood and your sleep, are all part of that broader physiotherapy sort of conversation. And that feels very much the way we run in clinic at King's. And, it's a very big picture. And obviously we're supported by our community therapists. And, in the gym, if patients are going to do a safe and managed program in the gym under a personal trainer, then we definitely liaise in with those people to make sure they're not doing harm, but they're sort of optimizing their function as long as they possibly can. And we're also there to support them when perhaps exercises in the formal sense are not quite the right thing anymore, but we help them in the next way that we can, in terms of somebody helping stretch them or passively move them, or just making sure they've got the right equipment, absolutely, as Nick said. So yeah, thank you.
Thanks, Jo. That's really, really useful. I think it feeds into one of the questions we had in advance, actually, was around how can someone access a physio with specific knowledge of FSHD? Does that have to come from a referral through the consultant, or what's the pathway to someone finding someone like you or Nick?
Well, I'll answer that from our perspective, and then perhaps Nick will step in. I think my great hope would be that everybody on this Zoom meeting today would be tapped into a local neuromuscular specialist centre, and through that neuromuscular specialist centre, they'll obviously have a consultant as their sort of primary lead for their care management. And I would very much hope that there would be a physiotherapist either in that MDT clinic with a consultant, or that consultant can direct them into. That's certainly the way we work at King's, and I'm sure that that's the way most of the specialist centres work. If if there's not that specialist therapist in clinic, then hopefully the clinic consultant will have an in-house referral system to the neurophysios in general in the outpatient department. And certainly it's a large part of my role, and I'm sure Nick's as well, to liaise with neurophysiotherapists who, whilst very broad and experienced, may not be so familiar with neuromuscular. And I would say nearly every week, a part of my job is either now a Teams call with the physio and maybe the patient, or a phone call with an osteopath, or a phone call with a Pilates instructor. So, and you know how you can access us specialist neuromuscular physios through the charity and through the network. So hopefully by facilitation one way or another, patients can get that specialist assessment.
Yeah, I've got nothing really to add to what Joe said. It's important that whoever it is, has a tie-in with a neuromuscular specialist centre, so they have the consultant, they have the support in that respect. And then the physiotherapist at the neuromuscular specialist centre would then act as an advocate and an advisor to whoever would be seeing them outside of that centre. We can certainly advise on types of exercise or types of activity that are going to be useful. But sometimes you need that additional support to be able to perform some of these tasks. And that's where we would act as that advocate and advisor.
Fantastic. There's a couple more questions related specifically to physio and exercise. So I'll stick with that theme before I bring Liz in, if that's okay. So several people have asked in the Q&A about hydrotherapy in particular. And I'm conscious that that's something that pre the pandemic there was already a bit of a lack of provision. And also we've had reports that hydrotherapy pools that have reopened are actually struggling to reopen, or not allowing to reopen to people who they consider might be vulnerable to COVID. So has there been a shortage of hydrotherapy? How important is it, and kind of how can people access hydrotherapy in their area?
I'll go first. My answer might be briefer than Nick's. We do acknowledge that for some people, hydrotherapy is very beneficial. We are aware, and I think MDUK have been aware, that there is a lack of facilities, and sometimes there's a lack of accessibility to our populations within those hydrotherapy pools, which is obviously, difficult and not what we would wish for. Some of our patients manage to access other public pools and might be part of, aqua aerobics, aqua yoga, aqua classes in more of a mainstream pool, which tend to be more accessible. I'm aware they're not as warm, and I'm aware sometimes the physical access isn't as optimal. So there is a shortage. I think we acknowledge that. I think it has become more difficult with the pandemic. I would also say that it doesn't work for everybody. Sometimes the sheer effort of getting undressed into the pool, out of the pool, dressed and back into their vehicle can be too demanding for some. So it doesn't suit everyone. I think all of us would wish there was more facilities and opportunities. Over to Nick for any additional comments.
Yeah, again, just reiterating what Joe said, access to hydrotherapy pools in general is very poor. We're very lucky here. We have a fantastic hydrotherapy pool, but at the moment it's only available to inpatients. It's not available to outpatients. Although when it does open up, we do 10 sessions for £28, and that includes the patient and the carer. It's open at the weekend, so they can pop along at the weekends. It's a completely accessible pool. But again, doing that once a week or once a fortnight, is that enough to actually promote functional change in muscle? I'd argue not. I think exercise or activity needs to be done on a very regular basis, once a day or once every other day, to actually improve that. You're not going to be able to get access to a hydrotherapy pool once every 2 or 3 days. So it's a problem nationwide for all conditions, not just muscular dystrophy.
Thank you. I promised to bring Liz in, and I will do next, but we've just had a question popped up that I think is quite important to cover. And I think probably while we've got James with us, James, you may be the best person to come to first, which was just simply how can someone get into a muscle centre? And I'm assuming that means get referred to a specialist. So just in case anyone's on the call who has FSHD or thinks they have and isn't in touch with a specialist, what should they do?
Yeah, I think it's a really good question, Rob, but it does highlight some of the problems we have in the NHS currently. So I mean, the usual, I guess, route of referral is people will initially see their GP, they'll then be referred to some kind of general neurology service, And then that neurologist will hopefully pick up that there's some kind of muscle problem and involve a specialist neuromuscular service, which may or may not be local to you. Some people do have to travel a long way away. And for some people, that's unfortunately just not possible, although we are doing more remote things these days. But if you're not with a neuromuscular specialist, I would look on MDUK's website. You can see where the bigger centres are that have neuromuscular specialists. You can ask your GP for a direct referral if you've already got a diagnosis, so you can ask for a referral to your local neurologist. And I think that's the route I'd take. The final thing I would say is that even if you get to a neuromuscular centre, what you might be offered is very variable between the centres, and the interaction between that specialist centre and your local services, so your community neuro rehab team or your local hydrotherapy pool, is not always an effective relationship. So things are very variable, and I think, as I was highlighting in my talk, these are things that we could really do with sorting out. And if we're giving people really sort of fancy, expensive new drugs, if they can't have a basic exercise program to go with it, you do wonder what's gone wrong, really.
Thanks, James. That's really good. I am now going to finally keep my promise and bring Liz in. Liz, you remain, unfortunately, a year after first saying this, you remain the last person I interviewed face to face for one of these sessions at an MDUK event, which shows how long we've been living in this virtual world. So just a quick introduction to Liz. Liz is an MDUK peer support volunteer and chair of the FSH support group, which is run through MDUK. So Liz is a passionate advocate and supports people living with FSHD following her own diagnosis in 2004. Liz, I don't know if you want to say a few words about the support group, and if I could tee you up with a couple of questions that I think you'll be able to cover. One was around advice for people who are seeking mobility aids, and also any advice around sort of home adaptation. So how can people sort of start understanding those two things? Are they a common query that come your way through the support group?
Yeah, and MDUK provide a lot of information on that. You've got an adaptions list, I think. I mean, I don't think you can actually specifically recommend a particular product just because things go out of date quite quickly. But obviously the support group is a Facebook group that does have hundreds of members who might have had experience of various things. So that, I mean, you can go through your OT to get support at home, just to have a look around and see what might be useful to you. You can self-refer in most cases to your local council's occupational therapist. I guess, like with mobility, that's quite specific to each individual because, FSH varies so much that you'd need to speak to the person on a one-to-one conversation to find out what their where they are with the condition and what might be appropriate at that level. So there's no one size fits all. I know that much. Every one of us is different. And the other thing, I mean, like with the registry, to go back to a bit of research, but what the registry questions will do is sort of maybe come to some kind of analysis of how long progression takes, and that's really useful for the farmers, I guess, come into the research, come into the registry for potential therapies. Yeah, but the peer support group, you can just ask anything, and if we don't know the answer, then we can point you in the right direction. Sometimes there isn't an answer, but sometimes just telling someone that you've got a problem is really useful. The muscle groups as well, they'll have lots of local information. So with it, when it comes to hydrotherapy pools and things, if there is one, then someone in the group will know about it and tell you how to access it. I know other charities probably run their own, bigger charities will probably run their own hydrotherapy pools. And I know some members have managed to get hydrotherapy that way via another sort of condition. So it's about, sometimes you have to the NHS isn't, is a wonderful service, but sometimes you have to kind of it's almost like you have to research yourself to find out what might be useful to you. So it doesn't, just because you're not offered it doesn't mean it's not there. So it's worth asking MDUK and well, with FSH, the support group as well, just to see if there's any local knowledge, someone knows something in your area. It's, you know, really useful for that.
Thanks, Liz. A question that came in sort of linked to something Nick and Joe said actually around the kind of the mental health benefits of exercise was someone actually asking about meditation as well. And I know that often we're asked in kind of muscle groups around kind of just the mental health impact of peer support. But do people come to you for any sort of advice about things like meditation or mindfulness or accessing sort of those kind of interventions? And be interested to hear from you, Liz, and then on to anyone else who's got a view on that as well.
Well, I mean, I sort of have my own mindfulness techniques, but yeah, I mean, you know, we would, I don't know if I've ever recommended going to someone about using a mindfulness technique, but I certainly wouldn't dissuade anyone from accessing it, you know. So yeah, I mean, I think with the support group, sometimes just, like I say, talking to someone else about something is just, you just feel, I don't know, listen to, support. I don't know, it's a weird, I couldn't explain why it works, but yeah, a lot of people just find, even though they know they can't do anything about their current situation, just talking about it and knowing someone else might be in a similar situation helps. But like I say, if we can offer a signpost to somewhere else that will help them, we do that.
Joe or Nick, have you got anything to add in terms of that element of living with FSHD?
I'm happy to go first, but certainly will then hand over to Nick. So I think depending where you are on your FSH journey, sometimes, you know, you can struggle a bit with your mood, you can struggle a bit with managing fatigue or managing pain or just managing the burden of having a long-term condition, which at the present time there isn't any sort of medical drug treatment for, although obviously we're all aware we have to support people in living well with the condition. And I think, you know, we would freely and happily enable people to either self-refer to talking services, and we can do that through in London through the IAPS. So people can self-refer for talking therapies. They might include CBT, and that might include sort of tools for the future in terms of mindfulness, meditation, other sort of relaxation activities. And I think all of us can benefit from mindfulness and mental health tools going forward, particularly having lived through a pandemic. So that would definitely be part of the bigger conversation within clinic, within the consultants or within myself. And I'm sure centres where you have the extra supportive psychologists or clinical nurse specialists or OTs. OTs are very, very strong in that type of support and, you know, recommendation with onward referral. So I would think, yes, there's only benefit to be had.
Yeah, I mean, we're very lucky we have a psychologist who works with the team. So if there's any major issues, we can certainly refer to the psychologist for help with mindfulness, CBT, etc. But I also think that being a physiotherapist is not just about the physical side of things. We also should be and advocate, and we should be the great listener. And sometimes I'm guilty of it. We always want to try and impart knowledge and try and tell people, yes, you should be doing this. But just listening to people and just letting people tell their story is really important, and just letting them, in a way, unload and unburden themselves is really important. And I think of physiotherapy as being sort of 60–65% the physical side of things, but also 30–35% is being the psychologist, trying to just listen to people and not giving an opinion, just listening to them. And that can just help. You know, they've got someone to talk to who's not a family member, who's just not in any nasty way bored or fed up with hearing the story again. And I know Liz is smiling there because it's the same story, but to me, it's not the same story. It's a new story. And I want to hear what your problems are. I want to hear you tell me about your life. I don't also want to come in to tell me, oh, I'm struggling doing this, or I've got some pain. I want to know what that impact has on you as a person, not just as a little, you know, a small microcosm of you. So I think we as physiotherapists should play a leading role in trying to help people's mental health.
Thank you. That's fantastic. I'm going to bring in Raj Badiani now, who is living with FSHD. So, Raj, you may have something to share on these issues we've discussed so far as well. But you're also going to tell us a bit about UK FSHD. So as I said, Raj was diagnosed around 4 years ago and is coordinating a group of patients, clinicians, researchers and patient groups to work together to accelerate therapy development and access to therapies in the UK. So yeah, Raj, feel free to, the floor is yours. Feel free to comment on any of the issues we've just been discussing. But yeah, great to hear a bit more about UK FSHD as well, please.
Thanks very much. Thank you to Kate and Rob and the team to give us this opportunity to share this work. It's the first time we are actually mentioning this. It's only literally just got off the ground, so to speak. Yep, I do have FSHD. I was diagnosed in 2016, and I think I can echo a lot of the things that I've seen on the questions, which is that it took ages to diagnose and all sorts of biopsies, etc. So there is work to be done in that space, and I think we shouldn't take our eye off it. But there's also other work to be done, which I think is pressing, given what we've just heard from Enrico and Linda, and also some of the stuff that Ben put up, which is around, you know, is the UK ready and coordinated to really act as a force with some of the work that's going on in the US and Europe? And that's what we're really trying to do here. All the people who work in FSHD are passionate about it, they're dedicated. So what we're doing here is really bringing that together in one group and trying to talk with one voice and also coordinate it. So if I can move to the next slide, please, Kat, thank you. So the idea seems really simple, and I think it has been tried, but perhaps timing, you know, people's time, the timing in terms of the therapies, etc., probably wasn't correct. And I think we're in a really optimum place now, given what you've heard from Enrico about the CTRN, the European work, the FSHD Society work. So what does this mean? You know, and I think what we're trying to do here is bring all the key stakeholders and the active players in FSHD. So the people who you've heard from, there are some others, and really try and see how we can accelerate therapy development and also get access to treatment. So what would make someone like Fulcrum or other companies, Dyne, etc., come here to the UK? What would drive them to talk to us? And this is our idea, that if we can coordinate together, but I think one of the things that James mentioned is really important, in that we don't take our eye off, and Liz has just mentioned that as well, that we do, you know, Nick, and around the therapy that's needed immediately for our people. So FSHD community will still need the care and support, but we do need a driving force that starts to look at how do we, UK, get ready for these clinical trials, for the therapies that are coming down the road. And they're not going to be here next week, we know that, but it's going to take us time to be ready too. So this is the idea that, you know, with patients, you can call yourself an advocate, or families who have members of their family who have FSHD will be one of the big groups in this kind of core team that I'm calling here. The clinicians, you've already heard from people like Enrico, James, etc. We need them. We need the muscle centers. We need the research scientists. Peter and Linda have talked about the research that they're doing. They feel a big gap around things we don't know, and we'll find out and will help us to push forward. The registry, which should be the red box there, I'm not sure why the text isn't there, but that should be the registry. That's sort of Ben, and we just heard Lindsay fill for Ben. The registry is absolutely phenomenal in terms of knowing what the areas of focus should be, who we are, where we are, what our particular necessities are. It forms a really big part. So I would really encourage anyone and everyone who can get their members of the family onto the registry, because that's going to form a big knowledge base. And Lindsay's assured us that this is very cleaned up data, it's anonymized, but how do, you know, it might help us make certain decisions about, well, where should the next CTRN be? This is a clinical network. How long would it take for our patients to get to that? So these are the kind of things that we want to think about strategically. One big thing to call out is that this is not a group on its own right. It is part and parcel of and under the guardianship and guide of MDUK, the FSHD Society, and the European network that Enrico mentioned. So we're not on a splinter doing this on our own. We're within the arms and embraces of these very established frameworks. and we want to work within that to make this happen. It's just how do we get together and become a recognized voice. And one of the big things is that how do we become easier to do business with? Because if we're all segregated, separated, it's very hard for someone like Fulcrum or another company to come to us. Same with our regulators. You know, if they don't have a voice from us, which is coordinated, it's very hard to know where the passion or the force is. And I think the dotted line you see there shows industry and regulatory framework. They're just some of our stakeholders. We'll talk a little bit more about them in a minute. I'm conscious of time. But this is the idea. We become a single voice, we become easier to do business with, and we are passionate in making this happen. Every single person who's joined this group is just dedicated to making this happen. So I'm very excited about it. So I hope you will be too. And if you're interested, we'll talk a little bit more about how you can get involved in this. So Kat, could I have the next slide, please? I won't go into this too much because I've sort of talked to it, but what's this team going to do? Drive a strategy, time-bound goals, get together, have a group forum to talk the single talk, so to speak, have a presence, be united in our patient's voice. And I think the regulatory framework and working with them is going to become really important. So if we can work out a framework that helps us to drive that, whether it's Fulcrum or Dyn or anybody else, and we're ready, that will help us to move faster and respond quicker. Thank you, Kat. Very broad timelines for this group. We're meeting for the first time on Monday. As I said, every single person who's on that hexagonal area, they are already working at full pace and full time limits, but they're dedicated to work with us in this area. And we have 2 to 3 representatives in every one of those hexagonal boxes. I put the task force there for Fulcrum. It's labeled as that, but really it's about how do we make a framework that we can use for any other company? How do we establish the relationships? Rob has excellent relationships with the regulatory forces. How do we use that to make sure that they understand what our pain is in living with FSHD and what it would mean for any particular therapy that comes along? And you know, get that through the channels to be of use to us in a year or 2 years. But how do we get those frameworks in place now? And we're not, you know, trying to do that at the last minute in 18 months' time when these companies do come knocking on the door. And what will make them knock on the door is something like this, where it's easier to talk to us. So by the end of sort of this year, I would really hope that we have 2 or 3 core teams set up, starting to have some goals. We're really trying to get the stakeholder engagement in place and a comms plan, which I don't know what that looks like at the moment, but I would hope that we can come back to this group, share ideas with you, get feedback, and also hopefully get engagement with the rest of the FSHD community through this mechanism as well. Thanks, Kat. I won't repeat this. You know, the farmers, etc., are knocking on the door. They're not far away. We need to be ready. You know, it's as simple as that. Ben shared the slide. This is from June in the USA. And if we start now, we can be ready, we can be organized, and we can have one voice. So I keep saying the same thing, but it's really the message of this group. Thanks, Kat. Oh, okay. That's taken a very unusual turn. This should really be the various stakeholder groups. So I think, Kat, we can probably move on. One of the things is that there are many stakeholders. The UK regulatory bodies are just one of them, the industry is just one of them, but there are other groups. Enrico mentioned the European network, there are other bodies. And it's important that they understand that we, as a UK FSHD core team, are working together to a strategy that is within the frameworks, as I said, of the European Network, FSHD Society, and then MDUK. And that, you know, we want to be ready to take on the therapies and the companies that are coming forward with those things very, very quickly. So if you are interested in being a part of this or would like to talk to us, please contact us through this email group. Liz, as you know, already supports the group, and together with her, we will work to make sure that we get any queries answered. We will need an extended patient group. almost every company and regulatory bodies need to hear from us in terms of what our pain is living with FSHD, and we need to get that documented and written in a way that really makes them understand what it means to be living with FSHD. So I hope you're excited. We are, to make this happen, and if you can be involved, please get in touch.
Thank you, Raj. That's fantastic, and yeah, looking forward to working with you. That brings us to the end of today's session, and I'm amazed we're only 3 minutes over, given how much we've managed to cover. So thank you so much to all of our speakers. I won't name you all individually, because then we will be 10 minutes over time, but thank you so much. I'm very conscious there are a lot of questions we didn't manage to cover. So if you had a specific question, the quickest thing to do is to email info@musculardystrophyuk.org, and we'll be able to help you through our advocacy and helpline team. We will go through the questions as well, though, and come back to people where we can, or put information on the website. We will also be putting a recording of this session on the website over the next few days, so you can revisit the slides and the information, etc. I'm going to end with a shameless plug, which is that a lot of the issues that have come out of this past sort of 2 hours were also exposed in 2 surveys we ran at MDUK, one of people living with muscle wasting conditions and one of neuromuscular services about the impact of an experience of COVID-19. We're holding a meeting via Zoom on the 21st of July of the all-party parliamentary group on muscular dystrophy in the afternoon of that Wednesday, where we'll be going through the findings of those surveys and also outlining some of our recommendations that feed into things like the UK rare disease framework and upcoming NHS reforms. So do look at our social media. I think a tweet went out yesterday with details of that meeting, but there'll be more info on that as well. And do consider joining us for that if you want to sort of discuss these kind of issues further. So thank you. I've now rabbited on to get us an extra minute over time, but thank you so much for joining us. Nick.
Yes. If there's any specific questions regarding physiotherapy and you want to email them over to myself, and I'm speaking for Joe here, and then we'll try and get them sent back to you so you can disseminate them amongst the, because I know I've got a list of questions here which, you know, I could talk for 25 minutes on exercise alone. So
yeah, thank you. I echo that comment by Nick. I'm happy to answer questions as well. We could do that together.
Oh, thank you. And we've got that on camera, so we'll definitely be coming back to you. But that's fantastic. Thank you so much. And I think that just goes to show how much the community sort of relies on the sort of great work that we're able to do between the patients, the patient groups, and the clinicians and the researchers. So these sessions wouldn't be possible without that dynamic we will have. So thank you so much. It's been a pleasure holding the session, and I do hope that those of you joining as audience have found it useful and informative. And take care. Enjoy your weekends when you get there, everybody. And I won't mention the football, but come on, England.
Thanks very much. Bye,
Thank
everyone.
you, Rob.
Thank you.
Thank you very much. Bye.